Bismillahirrahmanirrahim. First year, we will start the chapter number 3, that is enzymes. And before this, we cover the two chapters. Number 1 was the introduction of the biology, and number 2 was the introduction of the cell, the biological molecules. So this is the third chapter, enzymes, in which we study the structure, the properties, mode of action, and some important factors which affect the enzyme activity, as well as we will discuss the enzyme inhibition, inhibitors, and the last topic is the classification of enzymes. So this is all about the enzymes, the explanation of the enzyme. To some extent, you people already studied this topic in the grade 9, where we also discussed little bit all these topics, but some new topics are also present in this, like the inhibitors and the mode of action and the classification. So we will study it in detail in this chapter. And first of all, we will study or discuss the properties of enzymes, that what are enzyme and what are the properties of enzymes. So what are enzymes? Enzymes are biological catalyst. Why they are said to be the biological catalyst? Catalyst is any substance, any chemical compound that speed up. So enzymes are those chemical compounds or organic compounds which speed up the chemical reaction within the biological system, within the living organisms, as well as it also work in the laboratory, in ex vivo, which is also known as ex vivo or in vitro. Enzymes, why enzymes are needed? Enzymes are needed for the speed up chemical reactions, because thousands and millions of chemical reactions occur at a time in the living organisms, and these chemical reactions can complete without the addition of enzyme, but it will take much more time, so that it can't be significant for the living organism, or it can't give basic product at the time of requirement. So when enzymes are added, enzymes speed up that chemical reactions and they give the product at the time of need. So enzymes are very important. Without enzyme, there is no possibility of living organism or the life. What are the properties of enzymes? We will study it here. That you can see the first one property of the enzyme is given here, that it increases the speed of the chemical reaction, as we already discussed, that it increase and without being consuming the speed of the chemical reaction, enzyme will be remain in the same shape or in the same structure, having no change in the enzyme structure. Number 2 is enzymes are required in very small amount. Very small amount of enzymes can accelerate or catalyze a huge amount of substrate. It means that not so much quantity is required for the chemical reaction. And enzymes are highly sensitive to pH, temperature. If the pH increase or decrease, or if the temperature increase or decrease, it affect the activity of the enzyme. So they are highly sensitive to pH and temperature, which we will discuss later in detail. They are sometimes highly specific, that only that enzyme can speed up the specific chemical reaction and not the other one. Like sucrase, here the example is given that sucrase only digests sucrose. Sucrase enzyme only digests this chemical reaction, but not the other one. And sometimes their specificity become low, that like the carbonic anhydrase. Carbonic anhydrase combine oxygen with the hemoglobin, and it also responsible for the carbonic acid and bicarbonate in the blood, the formation of the carbonic acid and bicarbonate in the blood. So sometimes specificity become low and sometimes it become very high. The next property is that it work in the vivo as well as in vitro. Already we have discussed that it work in the living organism, internal parts of the living organisms, while it also can work in the laboratory like in a glassware or any other material. When we add the enzyme, we can do the same chemical reaction outside the living organism. And some enzymes are required cofactor for proper functioning, and we will study this property in detail in later lecture, that cofactor are the non-protein part, and some enzyme need that cofactor for their activity. Enzyme lower the need of activation energy. Activation energy is the energy which is required to accelerate or to start the chemical reaction. And if enzymes are absent, that activation energy will be much more. When enzyme is added to a chemical reaction, it speed up the chemical reaction, and so the energy of activation become lower. So that's why we say that enzyme lower the need of activation energy. And enzyme only speed up a chemical reaction, not affect the equilibrium of that reaction. It doesn't participate within chemical reaction, it doesn't react with the reactant, and no participation in their product formation. They just speed up the chemical reaction, the combination of the reactant and the conversion of the reactant into product. They don't participate in the formation of the product. So this is the last property, that we say enzyme only speed up the chemical reaction and have no effect the equilibrium of the chemical reaction. Now we will study the structure of the enzyme. Enzyme have a globular structure, and they are protein in nature. So according to the conformation of the protein, they are either the tertiary structure or the quaternary structure, with the exception of only one enzyme, that is ribozyme, responsible for the formation of the protein or involved in the protein synthesis. Without that enzyme, all enzymes have globular structure. In the structure of enzymes, there is a main site which is known as active site. Active site is used for the attachment of the reactants, which are known as substrate. The substrate which is to convert it into product, the reactant which is to convert it into product are known as substrate. So the substrate are binded first of all with the active site with the bonding like the hydrogen bonding, sometimes hydrophobic bonding, and they then enzyme then convert these substrate into product. Active site consists of two portions. One is binding site, in which the substrate bind, and the other one is the catalytic site. And specific amino acids are responsible for the binding and for the catalytic site. Like here we can see that this binding site can be present in the different areas of the enzyme when we open the tertiary or the quaternary structure. Like this is the active site, this amino acid is for active site, this amino acid is for active site. So different amino acids are present at different areas, but when it adjusts itself in quaternary structure or in tertiary structure, all the amino acids are brought together in specific area where it combines and they form the binding site, and this binding site is responsible for the binding of the substrate with the enzyme, while the other amino acid which change those substrate into product are known as catalytic site. A binding site has special shape, and it can only therefore attach the specific substrate to it. Like we can see that this shape is responsible for the binding of this product, this substrate only. If the shape of the substrate and the active site is not matching, then there will be no binding of those chemical compound with the enzymes. So enzyme have specific shape which is known as binding site. Another site in which specific amino acids are responsible for the change of the substrate into product is known as catalytic site. And enzyme have these two parts, are these two sites.